We are surrounded by hidden microscopic worlds filled with fascinating life forms. Thousands of microbial organisms live within a single drop of water. On Pond Life, we're going on a safari to explore the microbial wildernesses that exist all around us. Today, I'm out looking for a group of organisms that evolved over 2 billion years ago. These organisms were the first to live by photosynthesis. That's the process of using sunlight to make sugars, and then those sugars are used to power the cell. A byproduct of photosynthesis is oxygen, and 2 billion years ago, these organisms became so abundant that they completely changed the Earth's atmosphere. It went from one that was very oxygen poor to the oxygen-rich atmosphere that we live in today. These microbes are called cyanobacteria, and lucky for me, they're just as abundant today as they were 2 billion years ago. In fact, I haven't had to go very far at all to find them. I've simply walked out the door, I've crossed the road, and I can already see a pond that is teeming with cyanobacteria. You can find cyanobacteria all over the world. There are currently around 3,000 described species with different morphologies and habitats, from freshwater ponds to Arctic oceans. They even live in soil. As I wander around Central Park, I'm on the lookout for signs of cyanobacteria. It's cyanobacteria that are making this pond so green. If you look closely at this water sample, you can see that it's full of tiny floating green particles. Each one of those particles is a colony of cyanobacteria, and each one of those colonies is made up of multiple individual cyanobacterial cells living together. To find out what species of cyanobacteria is living here, I'm going to have to put it under the microscope. The cyanobacterium blooming in the lake belongs to the genus Microcystis. Microcystis colonies are made up of many individual cells suspended in a clear mucus. A colony may start as one cell, which divides to become 2 cells, then 4, and so on until some colonies are large enough to see with the naked eye. The colonies grow fast in warm summer waters, and when their numbers get dense enough, we call this a bloom. Many cyanobacterial species are bloom forming, and you can distinguish each species by their unique colony shapes. One advantage of living as a colony is that when many individuals live together, tasks can be divided among the members. We can see this in another cyanobacterium from the lake. This one belongs to the genus Dolichospermum, and among its helical colony, some cells look a little different from the rest. These specialized cells are called heterocysts, and they have given up their photosynthetic ability to focus on the task of absorbing nitrogen. They build that nitrogen into molecules that can be shared and used by all the cells in the colony, and in return, the other cells share the sugars gained through photosynthesis with the heterocyst. By dividing up these tasks, each is run more efficiently and the colony can prosper. Thousands of visitors walk through Central Park every day, mostly unaware of the many tiny dramas playing out all around them. With a microscope, we can catch a glimpse into these unseen worlds. The bloom in the next pond is dominated by a cyanobacterium from the genus Aphanizomenon. Aphanizomenon forms long, thin, filamentous colonies, and while at first they appear to only drift, Under time-lapse, we can see just how busy they are. There is a good reason to stay on the move. Cyanobacteria sit at the base of the food chain and are good eating for a number of small predators. The ciliate is a specialist cyanobacteria predator. It's from the genus Nassula, and it's using its sensitive cilia to feel out a filament, searching for an end. Once located, it begins its work, sucking in the cyanobacteria like a strand of spaghetti. As the cyanobacteria get ingested, the filament bends and breaks, allowing it to fit inside the rotund little ciliate. This process can take a little time, especially if the filament is particularly long. The ciliate keeps going, and going, and going, and going, and going, until the cyanobacteria are completely swallowed up. Delicious. When I'm out looking at these microbial communities, I often see things that I want to take a closer look at. And there's only so much I can do out in the field with my portable microscope. So when I see something interesting, I take a sample, and that comes with me back to the museum and into the lab. While I'm working with the pond water, I want to keep everything sterile. This is because I don't want microbes that might be growing in the lab or on me to end up growing in my lab cultures. Now that the microbes are out of the pond and in the lab, I also need to make sure they have everything they need to survive. The bottles here contain different types of growth media. Each medium contains vitamins, minerals, and salts that the microbes need. This big thing is a biosafety cabinet, and it's going to help me keep everything sterile. I wiped everything down with ethanol before it went into the cabinet, but it also has this wall of air that passes from this vent at the bottom right up the front. And that prevents any microbial spores that might be in the air from traveling through into this cabinet. At the same time, the cabinet is constantly sucking air up through a vent in the top. And that means that if any microbial spores do make it through into the cabinet, they get sucked up into that vent rather than landing on my cultures. I add a small amount of pond water to liquid medium, or spread some out onto an agar plate. That agar plate contains growth medium too, but it's been solidified by the addition of agar, a kind of jelly-like substance that's produced by certain seaweeds. Some microbes prefer to grow on the solid surface of the agar, while others grow better suspended in the liquid medium. I keep those cultures in a growth chamber. The growth chamber maintains the temperature and provides a constant amount of light each day. Cyanobacterial blooms can cause real problems. Some produce toxins that are lethal to many animals. I'm interested in the microbes that thrive here, organisms like these flagellates, and this Euglena algae, or this Collodictian, all living among the bloom. Over the next few weeks, some of these microbes will grow in numbers until eventually I can isolate and identify individual species from these liquid cultures and from the agar plate. I'm hoping that by growing and studying some of these microbes that are present in the cyanobacterial blooms, that we'll learn more about the blooms as communities, and that we can understand some of the things that are causing these extremely common phenomena. Even our biggest cities contain microbial ecosystems that are vibrant and complex. Growing these organisms in the lab helps me to understand just what thrives in this microscopic metropolis.